You are listening to People, Place, and the Ocean. My name is Jackie, and I will be your host throughout this podcast series that is inspired by the Gros Morne community-led marine atlas. With each episode, we will bring to life the pages of the atlas by learning directly from the people who help tell the story about Gros Morne and its surrounding marine environment. You can download your copy of the Marine Atlas by visiting ecologyaction.ca and visiting the Marine Planning page. In this episode, we are going to dive into some of the scientific work that has come out of the Bauman Bay Aquarium and Research Station. A lot of data from here is gathered from university students and professors from Memorial University, along with other experts from different environmental organizations. The Atlas captures a lot of this work in Chapter 7 and references different research projects. An example of a project that is referenced a lot is the CURA initiative, which stands for the Community University Research for Recovery Alliance. And on page 60 of the Atlas, there's also a distributional map of research on corals and sponges that dates back to 1975. Keep this in mind throughout today's episode, as we are fortunate enough to be speaking today with a professor that has been involved in projects like CURA, in addition to many others. Our guest today is an expert on corals and sponges and will help us to learn more about the uniqueness of the deep fjord environments that are found here in Gros Morne. So today I am here at the Bauman Bay Aquarium and Research Station, where I'm joined by Dr. Evan Edinger, who is a professor with Memorial University and is currently instructing courses here at the Research Station. So as we unpack different elements from the Gros Morne community-led marine atlas, Evan is here to speak more about the scientific research and how that research has been so critical to our understanding of the marine environment here. So thank you, Evan, for being here today. Can you please say a bit more about yourself?
So, first of all, I loved coming out here to teach. I teach geography, biology, and earth sciences at MUN. I would say coming out to teach in Bauman Bay is the best part of my year. Teaching a field course just beats the socks off of teaching in a classroom. And I would say a day in the field is like a week in the classroom or more. Because the students, they're immersed in the environment they're studying. It's surrounding them. They're learning how to do things, but also learning about the environment just by being in it. So you can't beat it. And Joe Robleski, who wrote one of the textbooks we use here in the class, it's called The Marine Wildlife of the Gros Morne National Park Region. He used to teach out here as well, and he was the one who said, we have the privilege of teaching young people. And it's a privilege because somebody trusts us to be in front of a classroom or in front of leading a group of students around in the field and to know what's important. So I'm grateful for the privilege to be teaching classes out here.
That book, Marine Wildlife by Joe Robleski, I'm definitely going to add that into the show notes because I want people to know about this book.
Okay, yeah.
And what is your connection to the ocean?
My connection is to the ocean. So I was probably the kid watching the Jacques Cousteau specials and thinking, I want to do that. So I did an undergraduate degree in biology with a lot of marine biology in it, and my master's and PhD were in geology because I also love geology, and I combined them by studying paleontology, and specifically paleontology of corals and coral reefs. So I'm a PhD student and go off to Indonesia with the intention of studying the shallow seas of Indonesia as a modern analog to the ancient shallow seas that covered much of North America. And I get there, and this is the middle of discovering the global reef crisis in the 1990s, which is continuing, of course. And so the environmental catastrophe that was facing Indonesian reefs at that time, driven at that point as much by pollution from land-based sources as it was by destructive fishing practices and the beginnings of seeing the impacts of climate change on coral reefs. So the environmental impact on those reefs became my thesis because that was obviously the much more important story to tell than comparing it with fossil reefs, which I did do one chapter on. And so my focus ever since I came to MUN in 2001 has been marine environmental science. And I've been privileged to be part of the environmental science program at MUN as well, which is a graduate program that has students at all the different MUN campuses. So main campus in St. John's, Grenfell campus in Corner Brook, Marine Institute. And in fact, there's an environmental science student here at the station this week. There's a number of students in the course I'm teaching right now who are in the undergraduate environmental science program at Grenfell campus.
Okay, so all of those students are here at the Marine Station with you taking courses.
Yeah, this year I'm teaching the first of 3 courses. Depending upon the year, there can be up to 5 undergraduate courses per year. And so this is called Marine Principles and Techniques. I call it the little of this, little of that course, because the students get introduced to a whole bunch of different sampling techniques and approaches to studying the oceans, but especially to the different habitats. And we're in this fantastically varied environment where there's so many different habitats to study in and around Bauman Bay.
Yeah. I think that's really interesting that you did your studies in Indonesia on the corals there, because they must look very different from what we would find here. I mean, we're talking about entirely different ecosystems.
They're entirely different ecosystems, and yet they're linked by corals still being fundamentally the same kind of animal. They're still cnidarians that have a stinging cell and they capture their prey. The difference being that in Indonesia, you're studying shallow water tropical coral reefs, and they have algae living in their tissue. We could say that they're farming the algae, and that's what provides most of their carbon needs. Whereas the deep sea corals that we have here in Newfoundland and Labrador, I should say cold water corals, not only on the deep sea, they're entirely heterotrophic. So they are plain old animals that just capture stuff to eat from the water column. So we call them filter feeders, but in actuality, many of them are hunting with those tentacles and they're catching something tasty that happens to float by.
Interesting. And what do you mean when you say that the species is heterotrophic?
So we just divide animals into being autotrophic. So a plant is an autotrophic organism; it makes its own food. Heterotrophic means it eats something else, or mixotrophic. And so in fact, tropical corals are mixotrophic. They combine the algae that they farm with hunting zooplankton at night. And so they get their carbon from the algae that they farm, and they get their nitrogen and phosphorus from the plankton that they hunt at night. If you wanted to think of a mixotrophic plant, a pitcher plant is a good example of that. Pitcher plants live in really nutrient-poor environments, and so they supplement the carbon from photosynthesis by eating bugs.
Okay, yeah. I've seen a number of pitcher plants in Grossmorn area.
Oh yes, we love them.
Now, going back to some of the courses that you instruct here, what kind of activities do you have the students do while you're here?
So the last couple of days we've been doing conductivity temperature depth profiles. So conductivity is the way that we measure salinity. And because Bauman Bay is a kind of an estuary, a fjord is a kind of estuary, and so they're measuring these vertical profiles of temperature and salinity in order to understand how the estuarine circulation works inside the bay. So that's their first assignment. They've done that one. And then yesterday we were taking plankton samples, and today they were analyzing those samples. And then another example is just walking around and looking at the intertidal habitats we have right here in Norris Point. Norris Point's got a lot of diversity of habitats, but we'll take them up to Salmon Point, Green Point, look at some other rocky intertidal environments. We also go up to St. Paul's and look at the salt marsh there. Some of the other things that we're adding to the course is a bigger unit on video analysis. So you guys have an ROV, which is fantastic. The poor man's version of that is a drop camera, and so we're bringing a drop camera from the Marine Institute out into the deeper waters, and also just a GoPro, shallow water drop camera that students will use. You don't have to have thousands of dollars. You can get a GoPro and put it on a little crash cage and take videos of what's living on the bottom. So that's one of the other techniques we're teaching the students about, how to both acquire the video and how to analyze it.
That's a really good idea. It's also cool that these students can now learn how to do that, and if they have their own camera, they could go out and capture footage anytime they want to. So we have in front of us the Grossmorn Community-Led Marine Atlas, and that is what brought us here today. So I want to actually reference something that's in this book. So in the atlas, we make reference to the unique environmental landscapes that are here in Grossmorn, and one of those being the deep sea fjords. You've mentioned this earlier on about the deep waters here and the fjord environment, so I'm wondering if you can talk a little bit more about that particular environment here and how it's formed.
Fjords are fantastic landscapes, and we all see them as tourist landscapes. That's how Norway markets itself. And if you look at the tourism advertising for Newfoundland, you're sitting on Air Canada, and there they are. They're showing Western Brook Pond, which is a fjord lake. So that's the view of the fantastic landscape above the waterline, but they're equally fantastic underneath the waterline. So a fjord is a drowned glacial valley, so it's where the glaciers flowed out, and then that valley that the glaciers carved was then filled with seawater. And it typically has a sill at some point, or it might have multiple sills, and that means that the circulation inside the fjord is in some way restricted. Estuaries in general are very, very productive environments because you have a lot of nutrients, and you have them in shallow enough water close to the surface that there's lots and lots of photosynthesis. And that estuarine circulation is constantly replenishing those nutrients that come from the ocean. They're not coming from the rivers, they're coming from the ocean, from deeper waters that get brought into that estuary. And fjords are a really good example of that, and Bauman Bay is a particularly good example of that. We have more than 100 fjords on the island of Newfoundland, and of course there are dozens more along the Labrador coast, and we have hundreds more fjords up in the Canadian Arctic. So Canada is very, very well endowed with this kind of environment. And at MUN we have a marine habitat mapping group which is focused a lot on fjords. So we've done that for nearly 20 years. The first student we had in that group worked on mapping marine habitats in Newman Sound over in Terra Nova National Park. And then we've also worked in Gilbert Bay. I have a bunch of students working up in Labrador now. And my current student, who is the TA for this course, is working on marine habitat mapping in Bay d'Espoir on the south coast of Newfoundland. So Bauman Bay and Bay d'Espoir might be my favorite fjords on the island of Newfoundland, because among those 100 fjords that we have on the island of Newfoundland, they're unique in different ways. Bauman Bay because it stays really cold all the time, and Bay d'Espoir because it's what we call a warm water fjord. So the deep waters of Bay d'Espoir are a balmy 4 or 5 degrees, as opposed to our -1 that we have down at the bottom of Bauman Bay. Or we think about that cold intermediate layer that is off the coast of eastern Newfoundland right now, which is why it's 1 degree off of Terra Nova Park today. So because that Bay d'Espoir has that warm water at the bottom, it's got a lot of the species of deep water coral that we normally would be seeing way off the coast on the continental slope. Now there's another twist to the story. In 2021, the scientific icebreaker that I often do research on, this is called the Coast Guard ship Amundsen, based out of Quebec, we went to the Makovic Hanging Gardens off the coast of Labrador. And this is a deep water coral environment on underwater cliffs that are not in a fjord. They're in one of these deep glacial troughs that cut way into the continental shelf. And so it's like having those shelf edge habitats way off 100 miles from shore, but instead it's only 12 miles from shore. And so the nearest town is the town of Makovic. And it was actually a place that an Inuit fisherman from Nain told us about. And so we followed up on that traditional knowledge from Joey Agnetok and mapped the area with multi-beam sonar, planned the ROV transects, and put the ROV down. So I had the privilege of being on board the ship when we did those first dives on it. So 2021, we've gone back there in the last couple of years, and there's still ongoing research in that area, including things like looking at the genetics of the corals and what grows around them, many different aspects of that Makovic Hanging Gardens.
That's really interesting to think about the corals as a garden as well.
It's another one of my favorite places.
There's so many different fjords here, and it sounds like each of them throughout the province are quite unique. I wanted to ask you, what is it about the fjords here, specifically in Bauman Bay, that are more unique to some of the other ones around the province?
Everyone's different. Each one has its charm, right?
Yeah.
So I talked about Bauman Bay, and it's staying really cold down in the east arm, and we can see those big differences between the east arm and the south arm because of the differences in depth, but also because of the differences in water temperature and the way that the water moves around. The south arm will warm up more than the east arm will. So everyone's different. So I can pick my favorites, but that's also just the ones that I know about. And if you ask somebody from Lapoil, that might be their favorite one,
Right.
for good reason.
Now, with these really unique environments and because of the colder temperatures, how does that contribute to the diversity in the area?
Yeah, we can think about diversity in a couple different ways. You can think about the number of different species in any one habitat, or what I like is the number of different habitats that are represented in a particular area. So during a 2-week class here, I can take my students to 14 different habitats if I wanted to. And those could be different parts of the bay, different parts of the intertidal, different parts of the salt marsh. Salt marshes are really rare in Newfoundland. Sand dunes are really rare in Newfoundland. I can take my students to a sand dune and look at coastal sand dune ecology. So to me, what makes the Bauman Bay environment such a fantastic place to teach is the number of different habitats that are represented in it. And the part that we don't get to do with the students very much is that really deep basin that never warms up, and those organisms that are much more like their Arctic cousins, or that might have been isolated from the rest of the Gulf for a while because they're in this deep basin that they can't really get out of without going into shallow water.
Okay. Can you speak a little bit more about the specific type of corals that we might find here?
In the waters of Newfoundland and Labrador, we have now about 70 species of cold water corals. And cold water corals, they're not only the stony corals that would make a typical tropical coral reef, and which are also the ones that build a deep water coral reef off the northeast Atlantic, so for example, the European margin. We also have a number of different gorgonians, and so we would call those sea fans. And we have soft corals and sea pens. Sea pens look like a feather pen, and they're another group of these cold water corals. And then we also have the actual stony corals, which in our waters are mostly solitary corals. So we call them solitary cup corals because some of them actually look like a cup you drink out of, or others look sort of like a fan. So among those 70 species of corals that are found, we divide them into those 5 major groups. The ones that are most tolerant of really cold water are the soft corals. And so when we're working up into the high Arctic, in the really far north, really cold waters of the Canadian Arctic archipelago, we find the soft corals, but we don't tend to find very many of the other species. And the further north you go, the fewer species and the fewer categories of corals you'd find. I had a student who worked in Bombay on deep water corals named Emil Colpron. I think I sent you his paper.
Yes.
And Emil had a number of different parts of his thesis, and one of them was he went diving off of Gad's Wall with Bob Hooper, who was the station founder. And it's very, very rich in soft corals. It's very abundant. It hasn't got the diversity of the other groups of corals that we find in deep water, or for example in Bay d'Espoir or the Makovic Hanging Gardens, where you have those other longer-lived, the sea fans, Gorgonian corals. So we haven't got that wide diversity here of different types of corals, but we have really, really high abundance, which is not surprising for fjords because they're so productive. Yeah. The other thing that Emil's thesis brought up, and this links back to your community-led atlas. Emil, his project was sponsored by the CURA project, which I'm sure you're well familiar with, the
Yes.
Community University Research for Recovery Alliance project led by Barb Neese. And so Barb O'Neese, who was a fisheries sociologist at MUN, retired maybe 5 years ago. Barb and I were his co-supervisors, and Bob Hooper was on his committee. And Emil went and interviewed fishermen in Port aux Basques, here in Bombay, and up in Port au Choix about what were the different deep-sea corals that they might have seen during their fishing careers. And the importance of that is the fishermen go into all sorts of different habitats. And you compare that with what the DFO research trawlers would find. So that's how DFO assesses everything, is to trawl. That's how we count fish to do stock assessment. It works very well for that, and we get bycatch of corals and other species along with that. And that's how the initial maps of deep-sea corals in our province were made, was from DFO bycatch. But they were also made from fishermen's observations. So on the east coast of Newfoundland, you can match up the DFO trawl data and the fishermen's observations, and they complement each other very nicely, and those maps largely coincide. Here in the Gulf, the DFO research trawls were showing one set of corals, much more limited diversity, and the fishermen had seen a much wider diversity of corals because they're going into different habitats. They're using gear like long lines or gill nets that can be brought into much rockier habitats that the DFO trawlers wouldn't go into. So the fishermen have seen a much wider diversity of corals in the waters of the Gulf of St. Lawrence than what the DFO Research Survey trawls found. That was a really important contribution that Emil made with his thesis. And so I'm thrilled to see the community-led atlas because there's this huge wealth of knowledge about our environment with the fishermen. Mm-hmm. And it's not a question of us taking advantage of that knowledge, but rather let's put that knowledge to the best use we can.
Yeah, and finding ways to integrate both scientific knowledge with local knowledge. It's really important. And I want to get back to that because it's a really important topic. And, you know, the main reason why we have something like the Atlas is to put all those different knowledge types together in one place. But first, I wanted to ask you, when thinking about corals and sponges, which is your specialty, what role do they have in the greater ecosystem? Why are they so important?
If we think about a kelp forest is a habitat, and it provides a habitat for a lot of other organisms. It's a biologically built habitat. And so there's invertebrates that live in amongst the holdfasts, and there are invertebrates that swim around or crawl around on the blades, and there's lots of fish in that habitat. And so it's a really, really rich biologically built habitat in shallow water. And deep water corals are doing the same thing in deep water. They're building these biologically built habitats. So we talk about coral forests. Now, those forests aren't as tall as a spruce forest. They'll typically be 1 to 2 meters, maybe 3 meters high for the largest corals. They're long-lived organisms. So the sea fans that we have in our waters can live up to 150 years, some up to 200. Depending upon where you go, the species have different longevities. But that means that if you remove them by trawling, for example, in the wrong environment, then it's going to take a long time for that habitat to recover. So they provide habitat for other things to live in. Sponges do the same thing. So sponges are also a very, very rich habitat. Sponges are harder to age than corals. So that's their importance. They're also, in addition to being important areas for diversity, they're also important areas for productivity. So typically they're going to go in areas that happen to have lots of planktonic productivity, and they won't concentrate that, but they will concentrate that biological activity because of all the things that are living around them.
Yeah, they're definitely very important for a lot of species. Now, what's one way that we might be able to recognize changes in coral and sponge habitats?
We will resurvey areas in some cases. And so we're looking at documenting the distributions. We use that from ROV footage as well as the DFO survey trawls or fisheries observer data. But then we want to look at what's happened to them over time. And so we have resurveyed areas off the Southwest Grand Banks, for example. So in 2007, we had our first major ROV expedition in Newfoundland and Labrador waters off the Southwest Grand Banks. And then in 2022, I was part of a group that went back there to resurvey some of those sites. I wasn't able to go on the ship that year, but my friends were there and went along the same exact track that the ROV took in 2007. One of the great things about ROVs is you have really good position data, and so they could follow that exact track and resurvey it. And so one of my students is working with my colleague at DFO to help analyze that data.
Okay.
Can't tell you the answer yet.
Yeah, I guess it's a long-term process.
Yeah.
What are some of the ways that your research methods have maybe evolved over time? And what are some of the ways that maybe technology has helped or not helped?
So I gave a talk at the Deep Sea Coral Conference in 2005. And have you heard the name Dick Hadrick?
No.
He was the head of the Ocean Science Center in Logie Bay for a number of years, fish biologist. And so he and I had a student who was working on deep sea corals, and one part of her project was assessing the fish that were found in the same trawls as the deep sea corals. So I described our methods at the conference. So I said our methods are crude and destructive because we are trawling through these environments. We're not trawling through them intentionally. This is DFO going out and counting fish. And so we document the bycatch, but then we're also documenting what fish species are caught in the same trawl. Now, here's the problem with that. A typical DFO trawl is 0.8 nautical miles long, about 1.4 kilometers. And we can't say where within that 1.4 kilometer path a particular organism was found. So we can say that on a gross scale, there's a spatial relationship between, or a statistical relationship between this species of coral and that species of fish. But we don't know what the biological relationship is. It's only from that trawl data. Another student who's graduated last year, and she's now a postdoc at MUN, a short one-year research contract, her PhD was about the relationship between sea pens, which is a kind of deep water coral, and fishes in the Laurentian Channel Marine Protected Area. This is research sponsored by the Canadian Healthy Oceans Network. So she took that ROV footage from the 2017 and drop camera footage from 2018 and looked at what fishes are around which species of sea pens. So interestingly, there's a lot of juvenile fishes, a lot more juvenile fishes around these sea pens than there are adult fishes. What are they doing there? Some of them are just hanging out. Others look like they're feeding. We know that redfish will lay their eggs around sea pens, and so you can find redfish larvae wrapped up in the what we call the leaves of the sea pen. It's like the edges around the polyp. But that's just one species that we've studied that with redfish. There's lots of redfish in these habitats, and especially a lot of juvenile redfish. So again, we're seeing that relationship between them. That's because we have the privilege of going back to the same place 2 years in a row, and then again we can look at a little bit of that temporal evolution. Things aren't going to change much over the course of 1 year, but we can see that relationship between the corals and the fish surrounding them.
Wow, that's really interesting. In the Atlas, there's actually a section where it talks about future research opportunities. So I'm wondering, is there anything you think should be focused on?
Everything.
Okay.
So I'll say everything because everything in the Atlas is important and everyone's going to bring their own expertise to it. And the fishermen have their expertise from being on the water and seeing long-term changes that a lot of them see. When Emile was doing his master's mapping the corals in the Gulf of St. Lawrence, they would tell him things like, oh, we go out to fish for shrimp in the Esquimalt Channel and that's where we're going to catch a lot of sea pens. They recognized already that the hauls where they would catch a lot of shrimp were also where they would catch a lot of sea pens. Same habitat, deeper water down in the channels. So there's all sorts of avenues to follow up on there. And following up, so what do we mean by following up? Well, part of that is keep listening to the fishermen because they're out there every day and continuing to look at how we can pair up fishermen's observations and scientific research based at DFO or the university. We're not trying to use one to validate the other by any means. Rather, it's we can look at the same kinds of questions from different approaches. Just like that corals and fish story, taking those crude and destructive methods from 2005, then matching them up with the more recent ROV footage to look at the exact relationship between those sea pens and those fish.
Yeah. Can you think of any other ways that fishermen's knowledge might be helpful in terms of getting a better understanding of our marine environment? I know there's a lot that can be said about that, but, maybe just in terms of corals and sponges.
There's so many ways to do that.
Yeah.
There are so many ways to do that. And often it will come down to the fishermen have an observation. Okay, so that's data, right? It's not an opinion. It's data. So the fishermen have an observation, and we want to try to figure out why that pattern that they've observed is occurring. And they might have reached a conclusion based on their knowledge and understanding of the ocean about why that's happening, but then we can go out and bring scientific tools to try to understand that why question. So I'd say that's perhaps one of the best ways to integrate those two, is to combine the much more frequent observation that the fishermen are doing all the time over many areas of the water with bringing specific tools to answer much more specific questions. So what we've done a lot in the past with regards to fishermen's knowledge and corals and sponges is the question of where to look, because fishermen know the water They know the water through space and through time, right? So they know where the best places to go, and they also know how they've changed over time, and they recognize those changes in what they catch, in the weather, and how the weather affects what they catch or what they don't catch, etc.
Yeah, that's a really good example. Now I just have one final fun question. What is your favorite coral species before we close this off?
Well, that's a hard question. I would have to say it's the bamboo corals. So bamboo corals, we call them. So keratoisis. They live in deep waters off of our coasts. Fishermen call them bones. So it's called a bamboo coral because it looks like a bamboo stem where you have the segments and then the joints between them. So this species grows as segments of calcite and then proteins in between them. And so they're long-lived species, but we can also use them to track oceanographic change, and they're beautiful. Wow. So let's not forget that the animals we study are beautiful.
Of course. Oh, I'm going to have to look up the bamboo coral later. Well, Evan, this has been a really incredible conversation with you. I've definitely learned a lot. I really did not come in here knowing too much about corals, but now I feel like I know a lot more. Thank you for all of this today. I really appreciate your time.
My pleasure.
In the next episode, we will continue our discussion with Dr. Evan Edinger and explore the relationship between research, economic opportunities, and the conservation of biodiversity throughout this region. Be sure not to miss the next episode and subscribe to the podcast now and follow along as we tell the story of Gros Morne, the people, place, and the ocean.